This is the Cytoskel Actin Cell Motility video. This is figure 17-34 from your textbook, and we're looking at the difference between Lamellipodia and Fibroblasts here. By the way, singular would be Lamellipodium and Fibroblasts, which are shown here, and then these are plural when we have an A at the end. So these are both cellular structures that form due to cortical actin, as in it's part of the cell cortex. So Lamellipodia, these are wide, ruffly sheets, and so they're shown with this kind of this wider area. And if you look over here on the actual image of a cell, notice that here we've got the edge of the cell, and then these wide, ruffly sheets that almost look like sails. Each of those is a lamellipodium. On the other hand, a Fibroblasts is this little tiny spike, and they're very thin, and so you can see an example of one of those right here. It looks like it's more about, from their drawing and labeling, it's more about where they are within the edge of the cell, but that's not true. It's actually about the thinness or the width of the extension of the cell membrane. Also, we call this front of the cell the leading edge, and that's assuming that the cell is moving in this direction. In general, the Fibroblasts and the Lamellipodium, I think of those as kind of cellular extensions to try to receive any signals. And so, for example, if an axon were growing out, then it might have these very wide areas to try to capture whatever signals are out there. And so that's kind of how I think of this. Again, this is the leading edge here where the cell is kind of moving towards, and the trailing edge is this opposite side that does not have very many Lamellipodia or Fibroblasts, or if any. Okay, let's talk about what kind of process this is. So we call this crawling. It's also known as motility or cell migration. Those terms are all used interchangeably. These are due to the actions of cortical actin, and this is a type of eukaryotic cell motility. This is a coordinated stepwise process, and it uses very specific cellular structures and proteins. And make sure that you know which things are cellular structures versus which things are proteins. So you know kind of what scope you're looking at. Proteins are going to be a lot smaller, and inside the cell, cellular structures are some feature that we can see, which is a lot larger than any individual protein. And it probably is being held together due to some specific proteins. This motility is considered an amoeba-like process. However, the sort of cell types that we're talking about in this example are fibroblasts. So the example cell that your book uses for this type of movement is a fibroblast cell, and they use it during wound healing, which is an important time for cells to be mobile so they can get to the proper location. So let's talk a little bit about what fibroblasts are. This is just kind of a side note so you understand some context. So fibroblasts, this is one type of cell that is in the connective tissue, and they secrete extracellular matrix during development. And we'll hear more about that structure later. Another sort of cell type that's present in the connective tissue is a neutrophil. And so you're going to see an example about neutrophils and how they have this type of movement as well. This is the cell motility via crawling. And to highlight what the different roles are of the components in this process, I've underlined proteins in blue, and I've underlined cell structures in green. So let's take a look at this figure. It's figure 17-33 in your textbook. And to get oriented, we have a eukaryotic cell that we're going to follow here. And you can see it's got a really nice slope to the shape of it. This is where the Lamellipodium or Lamellipodia are found. It's in this thinner area, and this would be where the leading edge is. And this cell, let's say it's a fibroblast, is going to migrate along the substratum. A substratum can mean a lot of different things. Here it just means a surface. And so we're not getting a whole lot of context about exactly what that surface is, so I didn't make any underlining. So the cell, here's the nucleus in brown, and then in orange they're highlighting the actin cortex. This is another way to say the cell cortex, although we've just called it the cell cortex so far. And as you know, the actin plays a key part in the cell cortex. And we would define this as a specialized layer of proteins, and so while there are a variety of different proteins and it's complicated, I would still call this a series of proteins more so than a cellular structure. Although the cell cortex is large enough that it's probably almost in a gray area of being a small cell structure. Okay, so let's check out step 1. That is when a cell pushes a protrusion out in the front along this leading edge. And we see that in this part of the drawing here. And this is due to actin polymerization. You don't need to keep track of all this right now about which end is protruding and whatnot, but you can see that the cell is starting to move out in this direction. And this is due to some tension which arises from the cortex. So that's step 1. Then step 2 and 3 are both shown in this next step here. So in step 2, the protrusions will adhere to the surface, and so here's some attachment points here. They're called focal contacts, and those focal contacts are made up of many proteins, including integrins. And so integrins, that's shown in blue, those are a transmembrane protein, and we're going to hear more about those later. So this is where adhesion takes place, and then once that adhesion takes place, then the cell will drag itself forward due to this tension and kind of gets up going some momentum and then gets some traction. And actin and myosin motor proteins will contract at the trailing edge of the cell, which is over here. And basically these are some of the motor proteins that are going to move the actin filaments. So like when I talked about before that the cell was kind of like a tent and actin was the frame and there were little motors that would move it back and forth. Actin and myosin, those are the motors. And then the cell can keep moving. This is just one step that it took by moving this one little spot forward from where it attached. And so we can start back up at the next step here where now we have another protrusion that forms. One thing I thought was pretty cool is that I just did a quick Google search, and it turns out that there's still doing a lot of research on the cell cortex and this tension. In 2017, there was an article published in Nature Cell Biology that had to do with surface tension, which is due to the cell cortex. Also, if you'd like to see a movie of a cell moving in this way, check out movie 17.7 from your textbook. It's a neutrophil, and it's chasing around bacteria.